Good day, grade 12 learners, and welcome to this video where we will be focusing on a previous control test for term 1 to assist your preparations as you are studying for 3 topics, namely DNA, meiosis, as well as reproduction. Before we start with the question paper, we will first have a look at a few important reminders that you need to remember as you are studying and preparing for your upcoming control test. So firstly, please ensure that the topics that you are studying for the control test are the correct topics, namely DNA, meiosis, as well as reproduction. Remember that reproduction was broken up into reproduction in vertebrates as well as human reproduction. So please make sure you know both parts of reproduction in vertebrates as well as human reproduction. Longer questions that could possibly be asked in your control test includes firstly DNA replication, transcription, translation. You need to be able to describe these. The process of crossing over and random arrangement of chromosomes that occur on the equator of a cell in metaphase 1 and metaphase 2. How does non-disjunction cause Down syndrome? Then in reproduction, you need to be able to describe the process of spermatogenesis, the process of oogenesis, the functions of the amniotic fluid, functions of the placenta, the menstrual cycle, which includes the uterine and the ovarian cycles, the secondary sexual characteristics that occur on the onset of puberty, which occurs in both males and females, and then lastly, the differences between the umbilical arteries as well as the umbilical vein. Please make sure that you are studying these paragraphs from your exam guidelines, because the exam guidelines is ensuring that you have the correct structure and format of your answer. So please make sure when you are studying DNA replication, transcription, translation, study these things from the exam guidelines. Spermatogenesis, oogenesis, you have these things in the exam guidelines. So please ensure that you study from it. Please ensure that you are studying these paragraphs from the exam guidelines. If your educators did not issue it to you, please, you can access it if you have to type in Google exam guidelines for grade 12 life sciences of 2021. It's the revised exam guidelines. Please download the document and ensure that you have it, because that is it has all the paragraphs and all the information that you need in each and every topic. So as you're studying the topics, you ensure you're going through that. If you have viewed previous videos that I have posted on DNA and meiosis, I've shown you the exam guidelines that you need to know for DNA and meiosis. I normally go through it at the start and at the end of the videos, so that is the information that I am referring to. We'll first start with having a look at questions taken from a section A of a previous control test to revise for our term 1 control test. So we're first looking at question 1.1.1. Please ensure that when you're answering question 1.1, it's going to be multiple choice questions. So only write the question number along with your correct option. For example, if you're choosing A as your answer, you write 1.1.1 and you're simply choosing A. Don't write more than one correct letter, because it will be difficult for the examiner to actually mark that. So there are 38 chromosomes in a somatic cell of a donkey. How many of these chromosomes are autosomes? Now remember that your autosomes are going to be your, normally if it's going to be in a human somatic cell, you have 44 autosomes and you have 2 gonosomes. So if it's in any other living organism such as a donkey and there are 38 chromosomes, because there are normally 2 gonosomes in any somatic cell, you simply take the total number of chromosomes and you subtract 2 from it to get the number of autosomes. So you're simply going to say 38 minus 2 for the gonosomes will give you the number of autosomes. Therefore, the correct option is going to be 36. It can't be 38, it can't be 19, can't be 44, because 44 is going to be in a human somatic cell. Therefore, the correct answer is going to be D. 1.1.2 says below is a list of steps involved in protein synthesis. The coding part of DNA unwinds and unzips. Peptide bonds are formed. Thymine nucleotide pairs off with adenine nucleotide on the template strand. One strand acts as template strand. mRNA molecule is formed. Which one of the following is the correct sequence and combination of events during transcription? The coding part of DNA unwinds and unzips. Is that a step in transcription? Yes. Peptide bonds are formed. No, they are formed in translation. Thymine nucleotide pairs off with adenine nucleotide on the template strand. In transcription, you don't have thymine nucleotides pairing that occurs in DNA replication. One strand acts as a template strand, yes, and mRNA is formed, yes. So we now need to arrange this in the correct order. So let's go to A. It says 1, 2, 3, and 5. 1, 2, 3, and 5. But we see that 3 is incorrect, therefore A is out. 5, 1, and 2. So 5, 1, and 2. No, that's not correct. 1, 4, and 5. So 1, 4, and 5. That is what we ticked. So is number 1 coming first? The coding part of DNA unwinds and unzips, yes. One strand of the template strand, yes. And an mRNA formed, yes. So C is possibly correct. Then D says 1, 3, 4, and 5. 3 was not chosen as a possible correct statement. Therefore, the correct answer is going to be C. 1.1.3 says which one of the following involves the development of the young inside the body of the mother and where it's nourished by the yolk. So oviparity, viviparity, precocial, or oviparity. So oviparity, viviparity, precocial, oviparity. Correct answer is going to be A, because they're saying the young is growing inside the body of the mother, but it's nourished by the yolk. So viviparity, growing on the inside, nourished by the yolk, ovoviviparity. The next question, 1.2 says give the correct biological term for each of the following descriptions. 1.2.1, the type of fertilization where the ova are fertilized inside the body of the animal is referred to as internal fertilization. 1.2.2, the structure in the head of sperm containing digestive enzymes is referred to as the acrosome. 1.2.3, the stage when secondary sexual characteristics develop in males and females is referred to as puberty. 1.2.4, the reproductive strategy in birds where hatchlings are helpless and unable to move and feed themselves is referred to as altricial development. 1.2.5, the division of the cytoplasm immediately after the nuclear division is referred to as cytokinesis. 1.2.6, the type of egg produced by reptiles that has extra embryonic membranes is referred to as the amniotic egg. In question 1.3, we're going to indicate whether each of the statements in column 1 applies to A only, B only, both A and B, or none. We're going to either write A only, B only, both A and B, or none next to the question number. Please make sure you comply to this instruction. So 1.3.1, discover the structure of the DNA molecule. Is it Watson and Crick, or is it Darwin and Mendel? Is it both, or is it none? We didn't learn about Darwin and Mendel yet. We only spoke about Watson and Crick. Answer is A only. 1.3.2, increases genetic variation. Does prophase 1 increase genetic variation? Metaphase 1, metaphase 2? Yes, they all do, because crossing over occurs in prophase 1 and random arrangement of chromosomes occur in metaphase 1 and metaphase 2. And both of these things are going to increase genetic variation in the gametes, thereby increasing the chance of survival in the offspring. Therefore, the answer is both A and B. The next question is focusing on DNA profiling. When a thief broke into a car, he cut his arm on the broken glass. Scientists extracted DNA from the blood found on the broken glass. They analyzed this DNA sample and compared it to the DNA from 3 suspects, P, Q, and R. The table below shows the result of the analysis for the DNA from each source. So you have blood from the broken glass, then you have the DNA of P, Q, and R. Firstly, what do the diagrams above represent? So this diagram represents a DNA profile, it represents DNA profiling. 1.4.2, which suspect is most likely going to be the thief? Now you need to take each of these DNA profiles, P, Q, and R, you need to match it along with the broken glass. So if we go to P, there's 1, there's 2, and there's 3. So we write 3 at the bottom of that. If we go to Q, there's 1, there's 2, there's 3, and there's 4. So I write 4 there. If we go to R, we're only going to have 1, you're going to have 2, so there's only 2 there. So you select the one that has the most matching bars. Please use your ruler and place it on these bars to make sure you get the numbers correct. You write the numbers at the bottom like I did, and then you get your answer. Therefore, suspect Q is going to be the most likely going to be the thief. 1.4.3, give a reason for your answer. So as we can see, the black bands of suspect Q's profile matches the black bands of the profile taken from the blood on the broken glass. So you have most of the bands of suspect Q matching that from the blood taken from the broken glass. 1.4.4 says provide any 2 other uses of the evidence above other than identifying suspects in criminal cases. So how else can you use DNA profiles? There was a number of things we stated: paternity testing, identifying dead bodies, finding missing persons, etc. 1.4.5 state 2 possible disadvantages of using DNA in a court of law. Please make sure you know this. So firstly, human error can cause the DNA evidence to be mixed with another person's DNA. DNA evidence could be planted to frame a suspect. And then lastly, the suspect could have an identical twin. And the identical twin will have an identical DNA profile. That takes us to the end of this video. In the next video, we will be focusing on questions taken from section B of a control test. So please stay tuned for the next video, which focuses on our section B questions. Thank you very much for taking your time to watch this video.